Our next speaker is Dr. Wesley Shoop. Wesley is a former professor of parasitology at Murray State University, and before that he did his PhD at LSU, and I was his lab student when he taught Zoology 1002 in 1979 spring. He's been the director of parasitology at Merck and Company and vice president and global head of animal health at DuPont. He's identified and photographed over 1,000 species of flora and fauna in Mississippi, some recently published in his book, Mississippi's Natural Heritage. And we have him in the gift shop. And he's going to speak about it's a photo essay. You'll see some of the things in his book. A photo essay of the flora and fauna of the Pearl River microcosm. Thank you, Wesley.
When Abby asked me to participate in this, I was looking over my collection of photographs, and I was kind of overwhelmed by it. I've tried to give talks like this before and reduce the 1,500 down to 6, and it never quite worked well there. So I decided to do something different today. I decided to choose one and go into some depth with you. The one I chose, I hope, represents the Pearl River ecosystem. And because what I'm about to show you is an organism you may never have heard of before. It's not on this particular slide, this collage that I put together. It's not in my book. But it is a very interesting organism that pretty much tells us what the Pearl River is all about. The organism is called Olearia marciani. It's in the phylum Platyhelminthes, which means it's a flatworm. It's in the class Trematoda, which means it's an obligate parasite. That is, you probably will never see it, but it's about 300 feet from where you sit right now down on the bluffs and down in the river plain. This is the life cycle that I'm going to spend most of the time talking about. It is one of the most complex, it is the most complex life cycle amongst the trematodes, and it is one of the greatest and most complex life cycles in the world. And it exists right here in the Pearl River. And when you look at it, you know immediately when you see all of the different stages there that you're going to have to deal with a vast, wet, and wild ecosystem. And that is what the Pearl River is all about. This life cycle is very complex, and I'm going to break it up into starting with the core. The core is an obligate 3-host life cycle. And if I get my cat's laser pointer out, it is comprised of 3 hosts, a definitive host, and we define a definitive host as one in which there is sexual maturation of the organism. We have then 2 intermediate hosts. Intermediate hosts are defined by us as a host in which the parasite enters and advances but doesn't sexually mature. And so we've got up here in the definitive host mammalian carnivores, and they can be bobcats, wolves, foxes. It can even be your house cat and your domestic dog. Then over here is our first intermediate host is a mollusk, a snail. It's always a snail in a trematode life cycle. Over here, then we have an amphibian, happens to be a tadpole. That is our second intermediate host. And so that is the hosts that are involved in the core. The one that we'll talk about now, though, I'm going to go through and show you each of the stages in these areas. The first one we're going to talk about is the adult that resides in the intestines. The intestines that I'm showing right here happen to be those of a bobcat. And those finger-like projections coming up are the villi that are in the bobcat's small intestine, the duodenum, the proximal part of the small intestine. And they are, what they do is they increase absorptive area. And we see the parasite then, the Olearia marciani right here. It's upside down here relative to this micrograph. It's upside down and it's scoop shaped. It's got a hind body, a forebody, and the forebody is the scoop, and it fits down perfectly on top of each one of these villi. It's perfectly adapted for that. Over here you see a light micrograph that I took, and up here you've got these lappets on either side of the oral sucker. Those lappets are now down into the mucosa, into the crypts of Lieberkuhn, and they produce all sorts of enzymes that digest the bobcat intestine, and then they take it in and ingest it. That's how they get their nutrition. The hind body up here you see on the light micrograph. This particular organism is hermaphroditic. It's got Hermes and Aphrodite both in the rear end. The large structures there are the testes, and then right there is the ovary. So if this organism finds itself alone in the bobcat GI tract, as this one is, it can still mate with itself, produce fertile offspring. The eggs are produced, they pass out into the lumen of the small intestine, and then the bobcat defecates them out. So the eggs are defecated in a wet and wild system like the Pearl River drainage. The eggs find themselves into water, but before that, here is one of the golden brown eggs. It's about 120 micrometers long by 60 wide. These are the villi of the bobcat intestine. As they migrate out the south end of a northbound bobcat, they find themselves into the water. They are unembryonated at that stage. About 20 days later, embryogenesis occurs. You can see a larval stage starting to develop. You actually see it in this higher mag shot right here. You can see an elongate larval stage being formed in there. It's got a pair of eye spots. It has eye spots because this is going to be liberated into the water column. Up on the north end of that egg is an operculum, and with hydrostatic pressure, that organism will burst out of the egg, and if I can get this thing going in the right direction, it will become free swimming. That is the miracidium. You might be able to make out some of the cilia on its side there, which propels it through the water. If you saw it under a light microscope, you might be forgiven for mistaking it as a protozoan. But it's multicellular. It's got the pair of eye spots because it's out in the environment, and it's got apical penetration glands. This organism does not at this stage have a GI tract or anything near that because it's non-feeding. It's got about 24 hours to find its next host. It's a hunter. And what it's going to do is hunt down a snail. And when it does, it penetrates the snail and immediately advances to the sporocyst stage. The sporocyst is this amorphous mass here of red. The brown that you see here is the digestive gland of a snail, and this is a squash preparation. And what is interesting about this is each one of these strands is an elongated miracidium, but then they reproduce. And that's a very odd thing to say, a larval stage reproduces, because what this was the original host in the evolution of this organism. And the actual reproduction, if you look at it, it's a form of degenerate sexuality. It's parthenogenesis. But it breaks down at some point, and that original strand then produces all these others. And you might make out this small little structure here, here, and I've dissected one out for you. This is the next stage we're going to talk about. It's called a cercaria. And it breaks out of the sporocyst, it breaks out of the snail, enters the water column again, and is free swimming. It's got a body here that is stains deeply red, and then it's got a tail right here, and it's really quite an active swimmer when it gets out there. This is an electron micrograph I took many years ago. They always reminded me of a flying dart. When it enters the cercaria, when it enters the water system, it is free swimming again, as the miracidium was. It's an infective stage as well, because it's going to be searching for a tadpole. It too has a very limited lifespan of about 24 to 36 hours to accomplish that fact. And when it does, it will penetrate the tadpole. Only the forebody that was stained red will penetrate the tadpole. The rest of the tail drops off and is lost. But when it does, it begins to grow into a stage we call the mesocercaria. This is a live picture that I took that I had retrieved out of a cottonmouth. They're very active bodies. It is basically the cercarial stage, although much larger. It's got a ventral sucker, an oral sucker, 4 large unicellular penetration glands there that secrete hyaluronidase, which will dissolve the tissue between cells. This organism is an infective stage within the tadpole, and with that hyaluronidase, it can migrate through tissues. Nothing can stop it. It's like a hot knife going through butter with those enzymes. When a bobcat or a house cat, as I've drawn here, if they were to eat that tadpole stage, you would think the mesocercaria might be liberated here in the stomach, and they just might go to the duodenum right there in the small intestine. But oh no, they leave the GI tract completely. They take 2 tracts, one directly through the diaphragm, one to the liver, onto the right side of the heart, and then to the lungs. At any rate, whichever path they take, they end up in the lungs. It takes them 2 to 3 days to get there. When they get to the lungs, they stay there for about 2 weeks. And they undergo a metamorphosis to the next stage. And that stage is called now a metacercaria. So we've got a cercaria, a mesocercaria, and now we have a metacercaria in the lungs. And it's kind of interesting because we've got a metacercaria in the lungs of this mammalian carnivore and the adult in the intestines as well. The metacercaria looks like this. You can tell it has changed fundamentally from what the cercaria or the mesocercaria look like. We're starting to develop this forebody here, that scoop shape that I spoke to earlier about the adult. We can still see the ventral sucker, the oral sucker. These are the vestigial penetration glands now. This stage will not use them any longer. Down here, this deeply staining mass is a genital anlagen. It will give rise to the testes and the ovary in just a few days. The hind body is just this nub at this stage. Within about 4 days, this hind body will shape out something like that, and the testes and the ovary will occur in them. This stage provokes quite the inflammatory response in the lungs. And when it does, it irritates the lungs. The lungs respond in expelling the metacercaria. They are expelled into the upper part of the trachea with cilia and coughing. It's coughed up, and we see something quite unusual. The same organism gets swallowed a second time by the same host. And it is at that stage that scoop-shaped forebody then attaches to one of those villi in the small intestine. It then grapples on to that villus. It hangs in 4 or 5 days. It matures to a sexual adult and begins reproducing. So that's the core of the life cycle. 3 hosts: a mammalian carnivore, a molluscan first intermediate, and an amphibian second intermediate host. But one of the interesting things about this is that tadpoles do not generally find themselves on the menu of a bobcat, a fox, or a coyote. So this happens to be a very weak part of the life cycle, but Ilaria has an answer for that. It is an auxiliary cycle over here comprised of innumerable different species of hosts. We call it the paratenic cycle. A paratenic host is a host in which a parasite enters, but it does not advance. It stays the same. So what you've basically got here is a food web in which the mesocercaria gets into it, and it goes through these trophic levels, and it hangs on until it can re-enter the cycle. One of these is then eaten by our mammalian carnivore up here. And the number of hosts and the length of times we have to remain vague on. I've done studies, I've transmitted these things through 10 different hosts, the same organism, it stays the same. The mesosaccharida never turns on its old age genes. Only if it gets in the right carnivore do those genes ever turn on. Some of these stages are probably produced last year, some of them 10 years before, some of them 100, some of them maybe 1,000 years old. There's no way to age them. They do not grow old until the right stimuli come in and trigger those genes to go to the next stage. Probably the best way to explain this is with that food web is if the mesosaccharida that are in a tadpole, if that tadpole metamorphoses into the adult frog, they stay mesosaccharida. If that adult frog then is consumed by a snake, the mesosaccharida are liberated, they go out into the tissues of the snake, they remain mesosaccharida. If the snake is eaten by a great white egret, the mesosaccharida migrate out into the tissues, remain mesosaccharida. Getting boring? If the egret is eaten by an alligator, the mesosaccharida move out into the tissues, remain mesosaccharida. Now, the alligator in Mississippi is one of the apex predators, so it generally does not get eaten or preyed upon until it dies. And then it is eaten by a whole variety of different organisms, possums being one that scavenge it. Guess what? The mesosaccharida move out into the tissues and remain mesosaccharida. And this is an incredible life cycle. One person pushed them through over 140 different species. They never changed. They never grew old. They stayed the same. It's really a phenomenal system that they have. The way I got involved in this cycle and the way I worked it out was because there were some human infections involving malaria as well. Some of them fatal. It's a highly pathogenic organism. And this young man, unfortunately, was a camper. He collected a bullfrog. He failed to cook it properly. He ate it, and it was his last meal. At autopsy, the mesosaccharida were found throughout his body, from the brain to the lungs, to the heart, to the liver, to the kidneys, to the tips of his toes. This happens to be his right testicle. And you can see some of the focal hemorrhages in several places here. And if you were to do a serial section on them, which we did, you can see a nice mesosaccharida sitting right in that testicular material. He had over 1,000 in him at that particular time. So now you see at least a major part of the life cycle. But the one that is really the coolest of them all is one we found in my doctoral dissertation. Big discoveries are oftentimes accidental, and this one was. I happened to be maintaining our cats in our vivarium, running the life cycle through them. I gave one cat a subclinical dosage just to collect the eggs at the out of the feces, and then run them through this cycle to get the mesosaccharida stage so that we could conduct the experiments we were running. But one time the cat turned out to be pregnant, and I took her off study, set her aside, and 2 to 3 months later I was doing routine fecal smears amongst all the other cats in our vivarium just to make sure they were healthy. One of the cats out of the 30 should have been passing malaria eggs. 6 were. And that made no sense to us. And I immediately thought I had contaminated the slides. But when I went down and I decoded the slides and looked at them, a surprise was waiting. One of the 6 was the female. The other 5 were her kittens. That pattern did not happen by accident. It immediately spoke that it was vertical transmission. That vivarium happened to be on the 7th floor of a modern biology building. These other stages did not exist on that 7th floor of that modern biology building. We had no way to explain how we got from one animal to the other at that level. I don't have the time to go into the studies, But what we found was something that is vertical transmission. We called it transmammary transmission. And that ought to clue you in that it is postnatal. And what we found at that particular time is that the mesocecaria, if it gets into, didn't want to go there. Well, if it gets into a bobcat, if that bobcat is a male, it will undergo that intestine to lung to intestine migration I told you about. But if that bobcat happens to be a female, she happens to be pregnant and she lactates, she will pass those eggs directly to her infants, in that case kittens, and the complex circular migration will occur there, eggs defecated and then return in the cycle. This is the actual part of the Alaria marciani life cycle that actually maintains it in nature. Not this right here. This has to happen, but this is the most important part. Just to give you one of our experiments to show you how effective this transmission was, I allowed that female cat to mate over the years, and she produced 6 different litters. Here are the kitten numbers here. You can see, as I spoke to earlier, in the first litter, all of her kittens were infected. So too all of the second litter, the third litter, the fourth litter, and finally in the 5th and 6th litters, we basically exhausted her of the mesocecaria. We basically sucked them out of her body. And I should have said, and I failed to, that I did not inoculate her with any further mesocecaria other than that first time years before. So that small subclinical infection that I gave her, she then infected over 20 animals afterwards directly. And that is something that is incredibly rare in our area of parasitology. So that is what happens off of this branch over here with a carnivorous mammal. But we also have over here in the paratenic cycle other mammals such as primates and raccoons. Same thing happens there, but with one subtle difference. These are paratenic hosts, so the mother passes them to the infants, but they remain mesocecaria, a true paratenic host. So you've got 2 different phenomena going on depending on where they are in the life cycle. But it too is 100%. In all of the hundreds of studies that I've conducted, if you inoculate a pregnant female, she's going to infect her offspring. It's that effective. Just a couple of closing slides here. These are mesocecaria that are in the mammary glands of a mouse. The mammary glands, you can see the alveoli over here, and a big fat mesocecaria right in the middle of them. There's the ventral sucker, the oral sucker, the penetration glands, the genital onlogon, the excretory vesicle. Up here you see some stroma or connective tissue. The mesocecaria can be quite pathogenic even in the mammary glands, and what you see here is a large granuloma, quite a bit of focal hemorrhage around them. In each of the alveoli, that's milk in each one of them that is being produced by the epithelium of that alveolus. And if you think about these alveoli, they actually come out of the plane and they begin to fuse with one another, and they create larger milk ducts and ultimately into a lactiferous duct that then exteriorizes onto the nipple of the female. And, whoops, pushed the wrong button. And speaking of nipples, here is one. This is a cross-section that I made, and you can see it's a lower magnification. So now the alveoli are quite small. Those of you sitting close can still see the milk in them. Here is one of the lactiferous ducts that I spoke to earlier in the mammary glands that move up and through the nipple. All of this is smooth muscle of the nipple. The outer dark purplish areas are the epithelium of the nipple, and the lactiferous ducts extend through and exteriorize there. And those of you with very good eyesight can see this little red structure right down. That's a mesocecaria. And it is literally one alveolus away from being in the lactiferous duct, and then one suck away from being in the intestines of a neonate. So with that, That is the Alaria marciani life cycle. It is one of the most complicated that we know. And the reason I chose it is that in order for that type of a life cycle to be carried out requires an ecosystem that is vast, that is wet, and that is wild. And there are very few of those remaining in the United States. We are fortunate to have one just a couple 100 yards from us right now, the Pearl River system. And when you find those, they are to be revered and respected, and in the end, protected, because there's not very many left. Thank you, Abby. Yes, sir.
This is in reference to the human fatalities, with, based on the reproductive and the maturation cycle. Did that camphor ingest 1,000 frogs?
Yes.
Okay, from the frog.
Yes, he ate the frog. I mean, we don't know how much of the frog he ate, but he ate enough to kill him.
Questions. You mentioned that the intermediate cycle of snail. How does that compare with the liver fluke?
The liver fluke is another trematode.
Same family?
It's not the same family, but it is in that group called the trematodes, and it goes through a snail, and then they produce a metasacharia too, but it's out on a blade of grass. And that's when the organic lawnmower we know as a cow comes along and consumes that grass, and that's where the cycle, that's a two-host cycle.
Second question. We know what the Alaria eats. What's his enemy? What eats him? The kill.
What kills Alaria? Praziquantel, made by Merck.
Can he live through the digestive fluid?
Oh yes, oh yeah, they go through the digestive fluid, just in what I showed you in the cat diagram. It goes through the digestive fluid twice. And the reason for that is we believe that Alaria once had 3 intermediate hosts. And the definitive host that we now see is a combined intermediate and definitive host. At one point, evolution is a patchwork. It's not an engineer that sits down and builds something de novo. Evolution is patchy. It adds on to what was already there. This particular organism, at some time in the past, that metasacharia had a mutation that provoked an inflammatory response. When it did, it angered the host. The host responded with an immune and inflammatory response, kicked it out, and it fell right into the hands of the parasite. Because it went up, it was already pre-adapted for surviving the GI tract, and it then went ahead and matured in the small intestine, and it went from a 4-host basic life cycle to a 3-host life cycle. That's how these things occur. Yes, sir.
Yeah, so when you're talking about the parasitic cycle, you mentioned that the Alaria's aging genes were not turned on.
Right.
Has that process been sort of studied from the standpoint of human aging?
No.
Nobody's.
No. The people that study human aging, I know some of them. My daughter happens to be one of them. And they choose simple systems. They choose Drosophila. They choose mice. They choose C. elegans, simple things that can be maintained in the laboratory. This cannot be maintained in the laboratory.